In last class, we have discussed the etiological characteristic of the pulmonary tuberculosis disease. In that, we have seen that there are some biochemical reactions are there, with the help of which we are able to easily differentiate between the atypical and the typical mycobacteria. And using such a biochemical reactions, we are also able to differentiate between the Mycobacterium tuberculosis and Mycobacterium bovis species. Now, in next to this disease, pulmonary tuberculosis disease that we have to see here, epidemiology of pulmonary tuberculosis. Now, this pulmonary tuberculosis, it is known from very ancient time. The evidence shows that the spinal tuberculosis, it is detected from the Egyptian mummies. So that time Egyptian mummies means it's a ancient, very ancient disease, which is also present at that time. Also, it is also detected that tuberculosis bacillus DNA, bacillus DNA, has been detected by molecular analysis in the mummy, dated before Christ, 1550 to 1080. During this time, it is detected that in some mummies there is a presence of tuberculosis bacillus DNA is there, and this pulmonary tuberculosis it has been known from many centuries and able to cause various most important human infection. This pulmonary tuberculosis it is known from many centuries and able to cause important human infection, and in its global prevalence it occurs throughout the world. It is not limited to particular part of the world. It occurs throughout the world. Because of this, it is also causing devastating illness. This illness is very serious. Because of that, that person, if not treated, that person may ultimately die there. And the effect of this pulmonary tuberculosis ultimately may result into the death of that person. So its global prevalence, its devastating illness or effect, and massive death which is caused by this pulmonary tuberculosis, it has been also called as a white plague, and it is also called as the captain of all the man of death, means captain of all the diseases which are caused to the human beings or man. Now, the majority of the cases and the deaths that has been occurred because of the pulmonary tuberculosis, they occurred in poor nations. Okay, mostly because of this, there, there is not satisfactory conditions among these poor peoples. They are not able to eat proper nutrition. There is no sanitation conditions, and because of that, this disease mostly affecting the poor peoples, and these poor nations, they are devastatingly affected by this pulmonary tuberculosis. Now, India is one of the countries which are worst affected by this pulmonary tuberculosis. Here, more than 40% of the populations in India, they are infected, and some 15 millions suffer from the tuberculosis in the country. Means there in India, 40% of the people they have suffered from infection of the tuberculosis. But these infections, because of the pulmonary the mycobacterium tuberculosis, they not resulted into the disease. But out of that 40%, 50 million people they suffer from tuberculosis in nation in our country, of which over 3 million are the highly infectious open cases. And half a million of peoples, among these 3 million highly infectious people, the half million peoples, they die from the disease every year in India, which is equal to 1 person in every minute. So that much devastating effect is caused by this pulmonary tuberculosis in India. Presently, over 13,000 new cases of TB and over 600 deaths occur each year in United States. Though it is a developed country, still this disease it is causing devastating effect in the United States. Now this pulmonary tuberculosis it still accounts for over 1.4 million deaths per year, which is almost 11% of the deaths due to the infectious disease. Whichever diseases which occurs to the human beings, out of that 11% of the deaths they occur only because of the pulmonary tuberculosis, and the rest of the deaths they occur by other ways. Now, 1/3 of the world population has been infected by Mycobacterium tuberculosis. When we compare our nation with the other world or whole of the world, it is estimated that 1/3 of the world population, which is the population now today in world, out of that 1/3 population of the world, it has been infected by the Mycobacterium tuberculosis. Okay, so now a close relationship has been detected between the tuberculosis and the HIV infection. Not only does the HIV infection reactivate the latent tuberculosis infection, but it also makes the disease more serious and renders treatment ineffective. So a close relationship between the HIV infection and tuberculosis has been detected, and which shows that this HIV infection reactivates the latent tuberculosis infection in those persons which already suffered from the pulmonary tuberculosis. And it also makes that disease more serious and make also the treatment ineffective against the TB, that particular pathogen of TB. Now in turn, this tuberculosis, pulmonary tuberculosis, helps the HIV virus by speed up the development of HIV infection to an active disease. Now there is a contribution between these two diseases, HIV infection and the AIDS infection. Here HIV infection and the tuberculosis infection, there is a correlation between these two. Here HIV infection reactivates the latent phase of the tuberculosis and make that tuberculosis disease more serious and treatment against the tuberculosis infection ineffective. While this tuberculosis infection favors the HIV infection to become an active disease. And one more complication which is developed there is that this situation make more serious. The third complication which is detected here that makes the situation more serious because that there is an emergence and spread of multiple drug resistance among the tubercle bacilli. This is another serious problem because whatever the antibiotic we are using today against the TB infections, these antibiotics they are effective in today's condition, but it might not be effective in future conditions. So we have to search for new antibiotics for the treatment against the tuberculosis infection. Now there is another characteristic which is transferred from between these Mycobacterium species, that is the multiple drug resistance, and because of that this bacteria become resistant to the various antibiotics, and that's why the treatment against the TB it becomes more serious. And this is because of this. In 1993, World Health Organization it declared this disease as a global emergency. This pulmonary tuberculosis, it is declared as a global emergency condition because of this contributions between these two diseases and the multiple drug resistant character which is spread among the mycobacterial species. Because of that, the WHO declared in 1993 the pulmonary tuberculosis as a global emergency condition. Now the worldwide tuberculosis infection is mainly caused by Mycobacterium tuberculosis, but along with this pathogen, Mycobacterium bovis and Mycobacterium africanum, these are the other two bacteria, mycobacterial species, they are also involved in causing infections to human beings and other animals. As you know that the Mycobacterium bovis, it is also this pathogen, this species is also capable of causing disease to human beings. Similarly, this Mycobacterium africanum, it is also capable of causing infections in the human beings. Here, the transmission to human beings, to humans from the susceptible animals and species and their products is also possible. The transmission of these pathogens to human beings from susceptible animals and their products, it can be possible. Here, through the milk, if milk is not properly treated, then that infected cow may give this bacteria into her milk, and from where it can be acting as a source to the healthy person. Now, with the advent of AIDS epidemic, there has been steady yearly increase in number of global TB cases. As the AIDS disease becomes epidemic, and because of that, there is also steady increase in the cases of the pulmonary tuberculosis in the world. Now, available statistic indicates that a close association between AIDS and the TB. Therefore, further spread of HIV infection among the populations with high prevalence of HIV infection is resulting in a dramatic increase of TB cases. As you know that there is a correlation between these two organisms and these two diseases. It helps them to spread between the healthy persons. And however, this Mycobacterium avium complex has become the most common mycobacterial disease in US in AIDS patients. In the US, in United States, those AIDS patients which are suffered from HIV infections, they have the tuberculosis infection, which is mostly caused by the Mycobacterium avium complex. It is a group of bacteria which causes this type of tuberculosis in US in amongst the AIDS patients. Now, human infection with Mycobacterium bovis is used to be a common worldwide, and in many advanced countries such as UK, it has been almost eliminated by using a regular tuberculin testing of herds and slaughtering of the affected cattle. Here in UK, those animals which are having this kind of infection, they have been tested for tuberculin. Tuberculin, it is a test which has detected the immunity against this pathogen Mycobacterium tuberculosis or Mycobacterium bovis. Means it indicates if tuberculin test is positive, it indicates that host has already suffered from the TB infection. That's why the tuberculin test is positive, and in UK such tests have been carried out in cattle, and if some cattle is shown the tuberculin test positive, that cattle has been killed. So in order to avoid the source of infections among the human beings, this cattle which is shown tuberculin test positive, they have been killed. Infections, it can be spread between animals through the aerosolized bacilli in moist cup spray. Here, this Mycobacterium bovis, it can be spread among the cattle, among the animals through the aerosolized bacteria which is present in aerosols in air, and this bacteria comes in the air because of the cough sprays. And a few infected cow, they may shed this bacteria in milk, and which is infectious in human beings when consumed raw. Means without any treatment, if such milk is consumed by human, then in that human, this raw milk may act as a mode of transmission through which this mycobacterium bovis bacteria can be easily transferred, and it may initiate the infection in that host. The primary infection, it mostly occurs in children and would occur in clavicle or mesenteric nodes, and from where it could spread to the bone, joints and other pulmonary sites. Human infection with Mycobacterium bovis could be prevented by drinking only pasteurized or heated milk. Here, person-to-person transmission of Mycobacterium bovis is very, very rare. But in Mycobacterium tuberculosis, person-to-person contact is very, very high. Because if any patient, any pulmonary tuberculosis patient comes come in contact with healthy person, in such condition, transmission may occur from that patient to the healthy person. So this can be acting as a source there. So this is all about the epidemiology of pulmonary tuberculosis. Then next part is the pathogenesis of pulmonary tuberculosis. How this pulmonary tuberculosis disease develops in a host. Before that, we have to see the source of infection. Here, the source of infection is usually an open case of pulmonary tuberculosis. Means those persons which are suffer from pulmonary tuberculosis and they remain free without any treatment. They remain in the houses. Then in house, that person act as a open case, and it may possible that from that person which remains at the house, from that person the infection may be transferred to the other person in that house. Now it is estimated that an open case of pulmonary tuberculosis in India may infect on an average of 25 contacts before death or cure. You can see. कोणाचा आवाज आहे ते आवाज म्यूट करा. Now you can see here, it is estimated that an open case of pulmonary tuberculosis patient in India may infect on an average of 25 contacts, mean 25 persons before death or cure. And other forms of tuberculosis, they are of much less importance in public health, but the pulmonary tuberculosis, it is most important. Now the mode of infection is the mode of transmission of this disease is by direct inhalation of aerosolized bacilli contained in the droplet nuclei of explorated sputum. Okay, whatever the bacteria that has been given by that open pulmonary tuberculosis patient in the environment during the coughing, sneezing, speaking, this person, open pulmonary case person, it gives out numerous droplets as many as 3000 infectious nuclei per cup. So this much amount of bacteria it has been given by that open case source, and from that bacteria comes in the environment, and in the environment it remains for 8 to 10 days under suitable condition in droplet nuclei. As you know that we have seen in etiology, this bacteria remains in the environment up to 8 to 10 days, and it may act as an important source to the healthy person, and from in that healthy person this infection is transferred or it is taken by the inhalation mode. If such an aerosolized bacilli, they have been inhaled by that healthy person, and this inhaled bacteria, they are present in proper amount, and then these bacteria, they can initiate the pulmonary tuberculosis infection in that healthy person. Now the dried bacilli, which is present in the dust, they are much less infectious among that of aerosolized bacilli. Dried bacilli, which is present in the dust after coughing, sneezing, or speaking process, whatever the hard drop, large size of drops comes out during this process, they falls on the dust, and where in that dust this bacteria, it is not remain there for longer duration. So this earliest infection and spread occurs among the household or other close or prolonged contacts of open cases whose sputum may have over 10,000 bacteria per ml. So this much amount of bacteria, if present in the environment around that healthy person, then this acts as a very important source through which infection can initiate in the healthy person. Now this pulmonary tuberculosis infections, they occurs rarely by ingestion, means eating process. So this infection cannot be transferred from, if that pathogen, Mycobacterium tuberculosis bacteria, it is present in the food sample or in a milk sample. From that, when there's such a contaminated food sample is ingested by that healthy person, the chance of development of infection is rare. Then chance by inoculation mode, it is also very rare. If from the wound that pathogen comes inside that wound sample, wound of that person host, then from that wound it cannot initiate the pulmonary tuberculosis infection. But the pulmonary tuberculosis infection is initiated when these bacteria, they have been taken by inhalation mode through the aerosolized droplet nuclei, where they are present in tremendous amount. Now the primary infection typically result from inhalation of droplets containing viable Mycobacterium tuberculosis bacteria from an individual with an active pulmonary infection. If some persons which is having pulmonary tuberculosis, and to that pulmonary tuberculosis person, a healthy person come in contact, then that healthy person may have this infection, this primary infection, typically through the inhalation of droplet nuclei which contains the viable Mycobacterium tuberculosis bacteria. And the majority of the inhaled bacteria, they are arrested, or they have taken down by the natural defense system of our upper respiratory tract. And bacteria reaching the lungs, they are digested or ingested by a special kind of macrophage cell. These are known as alveolar macrophage cell. Here, when this bacteria comes in the lungs, firstly they are attacked by our defense system, and in particularly the alveolar macrophage cells. These are the primary defense systems which attacks first on this bacteria, and there are several factors which also contributes on the onset of this illness. And these factors, they are the number and the virulence of the infecting bacteria. Host factors, they include genetic susceptibility, age, immunocompetence, stress, nutrition, coexistence of illness, and all these factors, they helps for initiation of illness among the healthy person. So the primary infection to a healthy person, it is occurred because of the inhalation of droplets containing viable Mycobacterium tuberculosis bacteria. And after that step, all these bacilli, they have been engulfed or digested or ingested by a special kind of macrophage cells which are present in our lungs. And these lung cells, these WBC cells or defense system cells, they capture all these bacilli and ingest them. But there is a characteristic that this bacteria, tubercle bacilli, they don't contain or secrete any kind of toxin. Okay, this bacteria is not capable of producing any kinds of toxin. The exact basis of their virulence is not very well understood, but it seems that it is related to their ability. Now, we have seen the steps. Firstly, this bacteria is inhaled by the healthy person, and this inhaled bacteria, they comes in contact with the first defense system cell, that is alveolar macrophage cells. Okay, up to now we have seen. Now in next step, what are the bacteria that has been ingested by these alveolar macrophage cells? These cells, macrophage cells, their normal function is to kill that bacteria inside the cell by using some enzymes. But this bacteria has a capacity that it survives. It survives there in that alveolar macrophage cell and multiplies inside the macrophage cells. And this is the virulence power of this Mycobacterium tuberculosis bacteria. These bacteria, they are not easily killed by our defense system macrophage cells, but instead of that, these bacteria, they multiply inside the macrophage cells, survive and multiply there. Now, various components of the bacillus have also been shown to possess different biological activities, which may influence the pathogenesis. Such activities such as allergy, immunity in infections. These are the other components which are also important here. Humans, they are able to mount an effective defense against the infection. Okay, all the humans, they are capable of mounting effective defense against the infection, as only a tenth of the infected develop the active tuberculosis. It is estimated, or it is determined, that our defense system, it is capable of containing this pathogen very well. Out of 10 persons which are infected by pulmonary tuberculosis bacteria, that is Mycobacterium tuberculosis, among the 10th, only a single person may develop the active tuberculosis. But the rest of the persons, they have the capacity, their defense system has the capacity to kill that pathogen by different mechanisms. But only single person is not capable of having the ability to kill that pathogen. So in that, there is a development of active tuberculosis is there. Now, whatever the immune system, our immune system functions and contain this infection or kill that pathogen, pulmonary tuberculosis causing pathogen, that is Mycobacterium bovis and Mycobacterium tuberculosis, with the help of a specific immune mechanism. That immune mechanism which we call here, which is effective against this bacteria, it is a cell-mediated type of immune response. This is the most effective way through which this bacteria can be killed in that infected person. So, out of 10, only 1 person develops the active tuberculosis, while the rest of the 9 persons, they are capable of killing that pathogen, that Mycobacterium tuberculosis and Mycobacterium bovis, by using this cell-mediated immune response. Now, in case of this pulmonary tuberculosis caused by Mycobacterium tuberculosis and Mycobacterium bovis, the humoral immunity, it doesn't play any kind of important role. Here, the main defense role is played by the cell-mediated immune response. And the key cell which is activated in this cell-mediated immune response is the T helper cell which contains a specific surface receptor, that is CD4 receptor helper cells, and which can develop along two different pathways. There is a Th1 or Th2, which releases cytokines such as interferon gamma, interleukin 1, 2, toxic necrosis factors, alpha, other exerting different biological effects. So, by these two ways, Th1 and Th2, the immune system kills this pathogen in 9 cases, while the one case may develop the active tuberculosis. Now, Th-dependent cytokinins activates the macrophages cells, resulting in the protective immunity and containment of the infection. While the Th2 cytokinins induce delayed type of hypersensitivity reaction among that infected person, but which results in the tissue destruction and progressive disease. In second case of immunity development, there will be not any protective immunity, but there is a destructive type of immunity is there, and which causes tissue destruction and progressiveness of the disease. The essential pathology in tuberculosis is the production of infected tissues of a characteristic lesion. Now, in lungs of that infected person, there is a development or a production of infected tissues of having a characteristic lesion. That lesions, they are called as a tubercle. And these tubercles, they are nothing but the avascular granuloma composed of a central zone containing giant cells with or without cessation, and a peripheral zone of lymphoids and fibroblasts. This is the characteristic of tubercles. These are the lesions which are produced on the lungs of that infected person. Okay, in that infected person, this tubercle type of lesions are produced, and these tubercles are the characteristic of the tuberculosis infection, pulmonary tuberculosis infection. And because of this formation of tubercle, the name tuberculosis is given to this Mycobacterium tuberculosis bacteria. The name of this tuberculosis bacteria, it is derived from the formation of this tubercle in that human's lung. And because of this characteristic lung and tubercle formation, this bacteria is given the name tuberculosis. Mycobacterium tuberculosis is name derived from this tubercle formation. Now, the tuberculous lesions are primarily of two types, exudative and productive. The exudative type of lesions is an acute inflammatory reaction with accumulation of edema fluid, polymorphonuclear leukocytes, and later of lymphocyte and mononuclear cells. This exudative type of lesion, it is characteristically composed of edema fluid. There is a presence of fluid like material is there, which is surrounded by polymorphonuclear leukocyte cells. Similarly, lymphocyte cells are there, mononuclear cells are there, and this type of lesions typically seen when the bacilli are present in large amount and they are virulent. And host response is more in the nature of delayed type of hypersensitivity reaction than that of protective immunity. Now, this exudative type of lesion, it causes destruction. Instead of, again, this exudative type of lesion, the immunity which is cell-mediated immunity which is developed, it is a Th2 type of immunity is developed. Instead of creating the protection, this immunity causes destruction, and that's why it is not a protective type of immunity. While the productive type of lesions, they are characterized predominantly having cellular associated more. This productive type of lesions, it mostly have the prominent cells there. which is associated more with protective type of immunity than the delayed type of hypersensitivity reaction. When productive type of lesion is produced, productive type of tubercle lesion is produced, then it contains only cells there. And these lesions, it is mainly associated with the protective immunity than the delayed type of hypersensitivity reaction. You can see here through the air, through the mouth or nose, the aerosolized bacilli, they have it comes inside the lungs, and where there is a presence of particular type of organelles is there, alveoli, and in that these tubercle bacilli, they are contained by these macrophage cells. They are ingested by these macrophage cells, but this tuberculosis bacteria, they are not killed by these macrophage cells, but instead of that, these bacteria, they multiply inside the this alveolar type of macrophage cells. Okay, and they survive there in that macrophages. Then there is a there is now in response to any kind of infection, our body gives a response. That response is known as an inflammatory response. So against this type of infection, one type of inflammatory response is given. Some signals they have been given by these macrophage cells. So these macrophage cells, they give some signals in the form of cytokines, and which brings about the large amount of different type of defense system cells at that point. Okay, where the more immune system comes there, you can see here, these are the macrophage cells which are having the tuberculosis bacilli inside them, and around that there is a presence of cells, polymorphonuclear cells, leukocyte cells, lymphocyte cells, mononuclear cells. They are surrounding these infected cells. Okay, and this part which is containing this type of cells, these are the tubercles. These are the tubercle lesions. And because of the production of this type of lesions and the bacteria which causes this infection, it is the name tuberculosis is derived from this tubercle formations. And if these tubercles they have been broken, then this bacteria can spread from these tubercles to other part of the lung. Okay, it can be spread like this. Same thing is there. This is the tubercles, where these are the different kinds of cells, and these are the cells which are containing this pathogen, tuberculosis bacilli, which is these are the tuberculosis bacilli. Caseous necrosis there, necrosis is there. There is killing of the cells is there. Epithelial cells are there. Multinuclear giant cells are there. Granuloma is there. Fibroblastic cells are there. So these are the various cells which are surrounding that bacteria, and this forms the tubercle. Now, depending on the time of infection and the type of response, the tuberculosis, the pulmonary tuberculosis, it is classified into two different types: primary and post-primary tuberculosis. These are classified into two types depending upon what type and the type of response which is shown there. And on that, they have been classified into two types: primary and post-primary. The primary tuberculosis is the initial infection by tubercle bacilli in a host. Now, in endemic countries like India, this usually occurs in young children. Means the primary tuberculosis in India, it occurs in the child conditions, in the child age, in children's. This infection, primary tuberculosis infection, occurs in the young children. In these young children, these bacilli, they are engulfed by alveolar macrophages, where it multiplies and gives rise to a superficial focus of tuberculous pneumonia, which is commonly located on the lower part of the upper lobe. And this tuberculosis pneumonia, subpleural focus of tuberculosis pneumonia, it is known as a cone focus. Now in time, this tubercle bacilli may change to cheese-like consistency and is then called as caseous lesion, which we have seen here. This is the caseous lesion which is produced in the primary infection. Now, in such lesions, if such lesions, this caseous lesion, it gets calcified, it becomes hard, then it is called as a cone complex, and which can be easily observed by producing chest X-ray. In that, you can able to observe this cone focus very easily. Here, in this primary type of tuberculosis infection, the hilar lymph nodes, they have been involved there. The cone focus together with enlarged hilar lymph node forms the primary complex. And this primary complex formation occurs about 3 to 8 weeks from the time of infection and is associated with the development of tubercular hypersensitivity. Okay, we can detect whether that person is tubercular sensitive or not. If it is sensitive, then some kind of infection, some kind of immunity developed in these persons also. Now, in majority of cases, these lesions, whatever this caseous lesion, or bone focus or bone complex that is formed during the primary infection, these lesions heals or cures spontaneously in 2 to 6 months, leaving behind a calcified nodule there. On these lungs parts, they form a calcified nodule. Hard nodule is formed there. Hard structure is formed there, where the bacteria is present at the center. Around this bacteria, there is a presence of different type of defense system cells are there. Okay, so in majority of the cases, these lesions, caseous lesions or bone focus complex, they have been healed spontaneously in 2 to 6 months, which is leaving behind there a calcified nodule. Now, in these healed lesions, as you know that few bacteria remain there in this calcified nodule, healed lesions, and they remain there for latent stage. But in a few, particularly in children, where the immunity is impaired and other risk factors they are involved, these primary lesions, okay, in such persons where the immunity of that child is very low, and other factors are also important there. Nutrition is not very well. In such conditions, age factor is also there, immunity is also there. In such conditions, these primary lesions may enlarge and cause miliary or meningeal or other form of disseminated tuberculosis. Okay, from this primary complex or from these lesions, primary lesions, the infection may spread to other part of the body or other part of the lungs, such as disseminated tuberculosis means spread of the tuberculosis into the other parts, other part of the lungs also. While the post-primary type of tuberculosis, it occurs due to the reactivation of latent infection. As I have told that after healing of the primary lesion, there is a presence of few bacteria in these healed lesions. And after healing, this bacteria may get reactivated in latent type of infection or any exogenous reinfection. If tuberculosis bacteria may also come from outside or other differ from the primary type in many aspects. Now this post-primary type of infection, it mainly affects the upper lobe of lungs. The lesion undergoes necrosis, tissue destruction, leading cavitation there. Lymph node involvement is not usual here, unusual. So, in post-primary infection, there is a reactivation of the latent infection which is present already there in that healed lesions or calcified nodule. And this, in later infection or in post-primary infection, this bacteria mainly affects the upper lobe of lungs. And the lesions undergo necrosis, which they also cause tissue destructions in the lungs, and which forms cavitation, means there is a formation of cavities there is there on the lungs. And these necrotic materials, whatever the tissue destruction that occurs in these lungs, that necrotic materials breaks out into the airway, leading to the exportation of bacteria-laden sputum. Now, you have seen that in TB patient there is a coughing is in tremendous amount is there. It is seen in tremendous amount. Why? Because whatever the tissue necrosis or lung cells they have been destroyed in the post-primary type of infection, they comes in the airway. So there will be blockage of the airways, and this blockage should be clear. That's why that person coughs very fastly. And during that, whatever the content that has been come in this airway passage that comes outside outside through the mouth as a sputum. And in that sputum, there is a presence of tremendous amount of bacteria is there, and which then may act as a source of infection to those which come in contact. Now, in immunodeficient, cavity formation is unusual. Instead, in those persons where there is a low immunity is there is a widespread dissemination of lesions in lungs and other organs. In persons where the immunodeficiency is there, like that of HIV persons, where there is immunodeficiency is there, in such persons, the cavity formation is not taken place in the post-primary infection. Instead of that, this pathogen through the blood spreads to the other parts of the organ of that host. Here, the incubation period is about 4 to 2 weeks, and the disease develops very, very slow. This tuberculosis infection development is very, very slow. You can see here, this is the process on which we have discussed here. This is a primary tuberculosis lesions, which can be easily observed by skin, that is tuberculin test or X-rays. And this is the whole process through which this bacteria can be spread to the other parts. This is the disseminated type of tuberculosis through the capillaries. It comes in the blood, and through the blood it spread to the other organs of the body. Now, this is the calcified nodules, which are tubercle bacilli, tubercles, where these bacteria are present. They can begin the latent dormant tuberculosis. They can reactivate the infection. This is the active tuberculosis. After latent dormant tuberculosis, they get activated. It spread to the other parts of the lungs. Now you can see this video through which we are able to understand few more points.
Tuberculosis, or TB, is one of the oldest and most common infectious diseases. About 1/3 of the world population is believed to be infected with TB. Fortunately, only about 5% of these infections progress to active disease. The other 95% of infected people are said to have a dormant or latent infection. They do not develop any symptoms and do not transmit the disease. Tuberculosis is caused by a rod-shaped bacterium, or a bacillus, called Mycobacterium tuberculosis. An infection is initiated following inhalation of mycobacteria present in aerosol droplets, discharged into the atmosphere by a person with an active infection. The transmission process is very efficient, as these droplets can persist in the atmosphere for several hours and the infectious dose is very low. Less than 10 bacilli are needed to start the infection. Once in the lung, the bacteria meet with the body's first-line defense, the macrophages. The bacteria are ingested by the macrophages, but manage to survive inside. Internalization of the bacilli triggers an inflammatory response that brings other defensive cells to the area. Together, these cells form a mass of tissue, called a granuloma, characteristic of the disease. In its early stage, the granuloma has a core of infected macrophages enclosed by other cells of the immune system. As cellular immunity develops, macrophages loaded with bacteria are killed, resulting in the formation of the caseous center of the granuloma. The bacteria become dormant, but may remain alive for decades. This enclosed infection is referred to as latent tuberculosis and may persist throughout a person's life without causing any symptoms. The strength of the body's immune response determines whether an infection is arrested here or progresses to the next stage. In healthy people, the infection may be stopped permanently at this point. The granulomas subsequently heal, leaving small calcified lesions. On the other hand, if the immune system is compromised by immunosuppressive drugs, HIV infections, malnutrition, aging, or other factors, the bacteria can be reactivated, replicate, escape from the granuloma, and spread to other parts of the lungs, causing active pulmonary tuberculosis. This reactivation may occur months or even years after the initial infection. In some cases, the bacteria may also spread to other organs of the body via the lymphatic system or the bloodstream. This widespread form of TB disease, called disseminated TB or miliary TB, occurs most commonly in the very young, the very old, and those with HIV infections. Tuberculosis is
So this is all about the